Good morning. Morning. Well, and let's start with the recollection, a little bit of our previous lecture. Do you remember something? Well, we declared last lecture that two people, two outstanding scientists, were nominated as Nobel Prizes for the development of the concept of brain structure, organization, and function. If anybody can say what are the names of these people first, and what is their contribution to the development of the concept of the brain? Please do look.
Yeah, so this those scientists were Camila Golgi and Cynthia Ramon Cajal.
What is their personal contribution?
Ramon Cajal's personal contribution is knowing that the brain is a structure consisting of separate cells. And Camila Golgi was, I think, one of the first who created a zero image brain.
This is constituent, yeah? Okay, right. And what is the technical contribution of Golgi? Technical.
Created a staining method.
Exactly. He developed a staining of sections with zero information. Okay. Well, and what else? And do you remember what kind of paradigm, what are the concepts are there for the development, actually, for the development of medicine, including neuroscience? There are three paradigms. Do you remember? What are the paradigms? Preventive medicine,
Oh, yes. Translational and
translational,
personalized.
and
Personalized.
personalized. Good. Great. Okay, and now let's continue our lecture. Sorry.
Well, in English, I guess the translation is not on for the online students.
Sorry?
I guess the translation doesn't work for online students, so maybe we should really try again
Yeah.
to put on the translation of the screen of online students.
I didn't catch. Well, today I think we should invite people in Zoom by Zoom. Yes.
Yes, but the classmates said that the translation is not on actually.
Yeah. And what can I do?
Can I try to?
Yeah. Please.
Make it again?
Yeah. Please, you're welcome. Actually, that's all my business.
Can I have a minute? I'm sorry for interrupting you so much. What am I doing? What? 19. Also, okay. Спасибо. And also the person is next to me. Oh. So, I guess, how do we should place the camera on the students receive? And you will put me, which is every video frames.
Yeah.
So, I will try to put that. It's. Okay. So,
I think when you do that, if you put this picture here,
I'm not sure if it's a possibility of this period.
in this position, it's really my back is really small.
I guess sorry. And I'm going to use that one some. Давайте я открою один правильный видео. Наш прописал.
Russell, we continue our lecture. Well, and Santiago, he proposed that we is of three compartments. The processes, dendrites, mainly dendrites. One action, only one action, and the cell body.
It's about that Charles Church.
No. He's a founder of synapses. But in the previous lecture, I have shown that Santiago, he looking at this cell impregnation, he proposed that the neuron consists, and that is true, consists of three compartments: dendrites, which are coming several processes, which are coming to cell body, an action, which outcomes from the cell body, okay, and the cell body with the nucleus. Well, and the next subject.
So, you mentioned cell body is the axon.
Yes,
And
look,
dendrites.
look. What is the neuron? Huh? Well, so there are several dendrites. There are two, but it might be more, many more. There is only one axon, and there is a cell body with a nuclei. Thus, the information is never lost. It is coming to the cell body via the dendrites, and it is distributed, is expanded one axon until the axonal terminal, like this. Okay. Well, so these are dendrites. This is a cell body, and this is an axon, which is terminated with an axonal terminal. Which gives rise to synapses. The synapses, that is a specialized contact between two nerves, and the existence of synapses. It was predicted many years before it was really discovered. It was visualized, and it was suggested by Sherrington in '87, and he was a Nobel Prize for this hypothesis. At that time, there was a hypothesis. Well, so according, and only many years ago, in the late 50s, my supervisors in France, academician Couture and Professor Taxi, they really discovered synapses in the electron microscope with a very high resolution. Because, And that was, as I mentioned, that was in the late 50s, in the early 60s. And then it has been shown that the synapses consist of two compartments. Presynaptic terminal, like this, with all options, and the postsynaptic neuron, dendrites or cell bodies. And therefore, the information within this specialized context is transferred due to chemical signals, which is released. The chemical signal which is released from the presynaptic terminal, and this chemical signal affect postsynaptic membrane of the target neurons. Well, so presynaptic terminals contain many secretory vesicles which contain chemical signals or neurotransmitters. They are accumulated in these vesicles. And after all, these vesicles, these neurotransmitters are released from the presynaptic terminals by so-called exocytosis. Exocytosis, it means that these vesicles are inserted into the plasma membrane, and the content of these vesicles is just discharged into the intercellular synaptic cleft here, okay? Well, so there are two major compartments. I remind you again that secretory granules contain chemical signals or neurotransmitters, and the synaptic cleft between presynaptic axonal terminal and postsynaptic dendrites or cell bodies. Well, and there are several kinds of chemical signals according to their chemical structure, unique chemical structure. First, these are neuropeptides. Neuropeptides, these are small segments of protein molecules. Proteins are very, very large molecules, okay? And these molecules are cleaved, giving rise to small neurotransmitters. Then there are classical neurotransmitters, monoamines or acetylcholine. And this is a very small molecule. And only a few neurotransmitters are represented by amino acids. Very, very small molecules. Well, so there are three kinds of chemical signals which we could consider as a language of interneuronal communication. Okay? Neuropeptides, classical neurotransmitters, monoamines, acetylcholine, and amino acids. Well, and the signals which is generated by neurotransmitters is registered, these neurotransmitters, they affect so-called receptors on the target neurons. Receptors, these are molecules which are very sensitive to certain neurotransmitters. And via receptors, the signal is transferred into the target neuron. And now we know that there is a lot of neurotransmitters, different neurotransmitters, which are responsible for different functions of the neurons, which are located on the postsynaptic membrane, on dendrites or on the cell body of the target neurons. But there are also receptors which are located in the presynaptic terminal, but in the same neurons. It means that the neurotransmitters, or in this case, this is serotonin or 5-HT, it is released from the axonal terminals and it affects the receptors on the same terminal, on the same neuron. What does it mean? This is a mechanism for auto-regulation. It means that the neurotransmitters, which are synthesized in these neurons, control the functional activity of the same neuron according to a feedback. What does it mean? Could you explain me what is the functional significance of this feedback? Could you imagine something? Well, so it means that if in the actual physiological situation, the neuron, it produces a lot of neurotransmitters, a lot of chemical signals, many more than this neuron needs. Well, so this neurotransmitter decreases the production of this neurotransmitter in the cell. Decrease. And on the contrary, conversely, if there is a deficiency of a neurotransmitter in the intercellular cleft, this neurotransmitter, they would stimulate, would promote the synthesis and secretion of additional molecules of neurotransmitters. Yeah, please.
Excuse me, I probably missed points. What kind of neurotransmitters do that? 5-HT or
Well, so at any neuron which secretes the neurotransmitters, there are the receptors exactly for this neurotransmitter. This is serotonergic neuron, which secretes serotonin of 5-hydroxytryptamine. This is a chemical definition, okay? Therefore, this presynaptic terminal is under the control by serotonin. But if instead of serotonin, there is a secretion of neuropeptide, any neuropeptide, therefore these receptors would be affected by neuropeptides, and they would control the secretion, the synthesis, and the release of neuropeptides. Okay? Well, so this is a specific, very specific control by the same neurotransmitters which are synthesized in this neuron. Please?
more or less?
So, it's simple.
Yeah.
Yeah, that topic is quite complicated. And this, every single neuron is controlled by only one neurotransmitter, or several.
Certainly no. Well, so it is, well, the functional activity of any neuron is under a lot of different chemicals, different neurotransmitters. And I will show you a little bit later that there are two types of interneuronal regulation. First, this is a synaptic neurotransmission within a highly specialized contact, okay, which is called synapse, okay. But this and the receptors are located in the presynaptic terminal, and there are different receptors for the same chemicals, which are located in the postsynaptic brain. Okay. Well, so there is a lot of receptors. There is a high concentration of these receptor molecules, but the same receptor molecules are located all through the membrane of the neuron, and there are almost all neurotransmitters, dozens or even hundreds, in the intercellular cleft, not only in the synaptic cleft, but in this cleft between different neurons. And therefore, the same neurotransmitters provide their action on the whole surface of any neurons. There is another way of transmission, and it is called so-called volume neurotransmission. Well, so there are two kinds of neurotransmission: synaptic, located neurotransmission within the synapses, and also the volume neurotransmission, when the neurotransmitters affect the whole surface of the plasma membrane. Clear?
Yeah, but when we say serotonergically, neuron or the cholinergic neuron, does it mean that it is activated specifically by dopamine or serotonin, or this neuron contains the receptors for other?
Yeah, well, if we say serotonin for a monoaminergic neuron, it means that this neuron produce monoamines like serotonin, because there are three types of monoamines: serotonin, dopamine, and norepinephrine. Well, so this neuron synthesize either serotonin or dopamine or norepinephrine. Okay?
Any of them?
Yeah, one of them.
Or only one?
One of them. What? So we say dopaminergic neurons, serotoninergic neurons, or norepinephrinergic neurons. And the same neurons, after synthesis of this neurotransmitter, they release it, they discharge it into the intercellular cleft, and these monoamines, they affect receptors and they provide so-called monoamine action on the target neurons. And this is also the case for peptidagic neurons. Well, so neurons which produce neuropeptides, there are hundreds or thousands of neuropeptides which we know and which can play a role of neurotransmitters. Well, so and they also affect specific receptors on the target neurons and also autoreceptors. Is it clear, more or less? Huh? Yeah? No problem?
Okay, simple neuron, there's only one thing, just one neurotransmitter, one neuropeptide is correct.
Well, so, and then I will explain you, I will answer your next question. What are the main populations of neurons and their characteristics? And therefore I will explain you that there is a number of neurons very different. There are pure peptidagic neurons, which synthesize and release only neuropeptides. There are pure monoaminergic neurons, but there are also mixed neurons,
Okay, that will do.
which can produce several neurotransmitters in the same neurons. And there are also neurons which produce neurotransmitters in tandem. Two neurons produce only one neurotransmitter. Well, so I will explain you what are the characteristics of all of these neurons. Well, so first, these are peptidagic neurons. Monoaminergic neurons. What are the functional?
I think you missed one slide.
Sorry?
I think you have missed one slide.
Well,
I think
before?
Yes, you've passed one slide.
This one, yes, wrong. You're right. Well, so first, peptidagic neurons. Well, so as I told you, these neuropeptides, which play a role of a neurotransmitters, this is only a small sequence of the large protein molecules. Well, so these neuropeptides, they are synthesized like a large molecule from neuropeptide, large protein molecules. It is synthesized in the cell body, only in the cell body. Nevertheless, there are very recent, and it was, for decades, it was generally accepted that there is a synthesis of neuropeptides in dendrites and in cell bodies which are related to so-called ribosomes. Ribosomes, these are subcellular structures which play a role of a sort of template of matrix for the synthesis of protein. Well, so in these ribosomes, they are contained in dendrites and in cell body, and therefore it was considered for a long time, until recently, that peptides and proteins are synthesized only in dendrites and in cell body, but not in the axons. Because in the axons, there are almost no, or even no, absolutely no ribosomes. But only very recently, it has been demonstrated that there is also peptide synthesis in the axons, but at a very low level. It means that the majority of protein molecules still are synthesized in the cell bodies and in the dendrites. Huh? And then they are accumulated in small vesicles, in secretory granules in the cell bodies. And the secretory granules containing protein molecules and neuropeptides, they are transferred to the axonal terminals. And in the axonal terminals, they are released, these neuropeptides are released according to exocytosis. Exocytosis. And neuropeptides, they play a role of neurotransmitters in the synaptic cleft. It's not very good, very well visible here.
If you want, I can move the wheel screen.
Yeah, yes, it's possible, but. What should we expect?
No, I will try to place it somewhere here.
Okay, well, so neuropeptides, they are released via exocytosis from the presynaptic terminal into the intercellular cleft, and they affect receptors on the target neurons. And it is not shown here, it's not shown here autoreceptors, but believe me that all neurons, they have also autoreceptors for the same neurotransmitter.
Excuse me, could you repeat the last sentence? How do you call them autoreceptors?
Autoreceptors. That receptors of the same cell. Auto. Okay. Let's go there. I don't know why it doesn't move.
What do you
Listen, it doesn't look anymore. I cannot change.
I
Slide.
think you have to click on the screen of the mouse because it's moved to the application.
Look here.
Right now. Just click once on the screen and then it will work. Yes.
Yes, it works. Okay. Well, so the majority of the neurons are represented in the brain, are represented by peptide neurons. And there is a lot of neuropeptides, dozens or hundreds or even thousands, which play a role of a neurotransmitter. There are less neurons which are called monoaminergic. And this I repeat again that there are two kinds of monoamines: serotonin, dopamine, and norepinephrine. And the major characteristic of this, of these neurons, that they contain a number of functional molecules which are involved in dopamine synthesis and release. These are enzymes of dopamine synthesis. Well, so enzymes, they are proteins which are synthesized, as I mentioned previously, in the cell body or in dendrites, like all other proteins. Okay? And for the synthesis of monoamines, we need enzymes, because this is an enzymatic reaction, multi-step reaction of monoamine synthesis. Well, and so they are synthesized in the cell body. And these enzymes are distributed all through the neuron. I mentioned previously that neuropeptides are synthesized only in cell bodies and in dendrites for 99%. But in contrast to neuropeptides, monoamines are synthesized all through the neuron. In cell bodies, in the axons, and also in axonal terminals. And you can see these molecules which are synthesized all through the neurons. Well, so this is an example of dopamine synthesis. One of the most important monoamines, dopamine synthesis. Well, so for dopamine synthesis, we need two enzymes. Tyrosine hydroxylase, which converts a tyrosine, this is an amino acid, to precursor, L-dopa, okay? Tyrosine hydroxylase. And then L-dopa is converted with another enzyme, aromatic L-amino acid decarboxylase, into dopamine, the neurotransmitter. Is it clear? No problem? Okay, good. Well, so dopamine is synthesized all through the cell, in the exoplasm, in the cytosol, and then it should be taken. It should be isolated into it should be captured by so-called secretory granules. And there are two reasons to separate, to capture dopamine into the secretory granules. First, dopamine, if it is accumulated in the neuron, in the cytosol, in the matrix of the neurons, it becomes toxic. And it provides the death of the neuron. Therefore, the excess of dopamine should be isolated within the granules. And for these, there is a specific transporter molecule in the membrane of the secretory granules, which capture dopamine and other monoamines into the secretory brain. Well, so this is vesicular membrane transporter type 2. Well, so this molecule is responsible for taking up of dopamine and any other monoamine into the secretory granules. Okay? And then the secretory granules are transported into the axonal terminals, and they are released via exocytosis into the synaptic cleft. And after the release into the synaptic cleft, the dopamine and other monoamines, they affect receptors on the target neurons and autoreceptors. These are autoreceptors, and these are receptors on the target neurons. But in contrast, and after that, the excess of dopamine is destroyed with specific monoamine degrading enzymes, mainly monoamine oxidase. Well, so after the release and after the action on the receptors, the dopamine is destroyed. The great majority of dopamine is destroyed with the specific enzymes, monoamine oxidase. But still, the rest of dopamine, which still is preserved in the intercellular cleft, is taken up by the same neurons due to dopamine membrane transporters. Well, so in the membrane of the axonal terminals, there is a specific functionally important molecule which is called dopamine membrane transporter. It is responsible for capturing dopamine from the intercellular cleft into dopaminergic neurons for reutilization. This is a sort of mechanism of adaptation, okay? The reutilization or the use of many times of the same molecules. Yeah, please.
Is there a specific reason why some of the excess dopamine produced, some are reuptake back to the presynaptic terminal and some is destroyed? Like,
Well, yeah, I understand. Yeah, I got your question. At first, also after the action, after the release of dopamine into the intercellular clefts, there are several types of its action. First, on the receptors on the target neuron, this one. Another one, the action on outer receptors, just for the control of the of dopamine secretion by the same neurons. Okay? And then if there is an excess of dopamine in the intercellular cleft, it is eliminated by two mechanisms. First, it is destroyed by specific degrading enzymes which are located in the intercellular space. And it is called monoamine oxidase. Okay? And another mechanism is capturing of dopamine from the intercellular cleft into the neurons due to so-called dopamine membrane transporters. That's okay? Understand? Good.
But I was asking why maybe okay, so there's excess of dopamine produced in the cleft. Why not destroy everything or reuptake everything back?
Yeah, because that's very logic from the point of view of the nature. Because if you can use something twice, it's better if we use it only once. Okay, well, so we don't need to synthesize again a lot of molecules of dopamine. Some of them are captured and can be reutilized. You know, this is, I told you, this is a sort of mechanism of adaptation, compensation adaptation, right? Yeah.
Maybe I can reorganize the question. Why the initial enzymes create the excessive amount of dopamine? Or what happens and how do we can is there a mechanism to control the amount of enzymes that, or the amount of dopamine produced by enzymes?
Yeah.
And also, is there a mechanism to control the reuptake of this excessive dopamine in the intracellular space?
Well, so there are several possibilities that some neurons under certain physiological conditions are highly activated. For instance, dopaminergic neurons are responsible for motor function, for the regulation of motor function. And therefore, if we make physical exercises, morning exercises or so, these neurons are activated. Well, and there are some signals which are rising from the periphery of the bodies, and they stimulate the activity of dopaminergic neurons. But after all, when you are sleeping at your lecture, like some of them, well, so you don't need any more activation of these dopaminergic neurons. Okay.
Okay.
And therefore, there are several mechanisms for the diminution, for the decreasing of the activity of these neurons. First, these are autoreceptors. And via autoreceptors, dopamine decreases the functional activity and secretion of dopamine. First one. Another one, these are enzymes. Enzymes, they're proteins. If there is an excess of dopamine, there is an increase of two parameters. First, this is the activity of enzymes, because the same molecule of enzyme, it can be active, enzymatically very active, so it converted, it can degrade a lot of molecules, and the activity can be very low, and it can degrade only a few molecules. Okay, well, so the activity will be decreased, and also the number of protein molecules also can be decreased. Well, so there are two mechanisms to decrease the synthesis of this monoamine oxidase as a protein, and also to decrease its enzymatic activity. Okay? And these mechanisms of promotion and of decrease of dopamine secretion are interconnected.
I also have a question about the toxicity of dopamine. So this secretory granule, it is created by the same enzymes or by others, and what happens if they dissociate? And does it cause any, I don't know, something like Alzheimer's or peripheral?
Well, good question. Good question. Look, I told you if there is at a certain high concentration of dopamine, which is distributed all through the neuron in cytosol, in the matter, it becomes toxic. And it promoted death of the neurons. And you are quite right that this happens in Parkinson's disease.
I was.
In
Right.
Parkinson's disease, because in Parkinson's disease, dopaminergic neurons degenerate. And dopaminergic neurons, I mentioned already, this is a key link of the control of motor behavior. Okay? Well, and to protect this dopamine toxic action, dopamine is captured by vesicular membrane transporter type 2 into the vesicles. And in the vesicles, it is not anymore toxic. Okay?
But as I know, Parkinson's disease is widely treated with L-Dopa at first, and then the efficiency of this treatment decreases. But why? Why does it happen?
Well, sure. Exactly. This is a problem because since, as far as I remember, since 60s, all the patients with Parkinson's disease, they are treated with a precursor of dopamine, L-Dopa, just to compensate the deficiency of dopamine because of the degeneration of dopaminergic neurons. In Parkinson's disease, there is a high degeneration of dopaminergic neurons. And therefore, for this reason, there is a deficiency of dopamine, local deficiency of dopamine. And therefore, it is very logic as a treatment to compensate this deficiency. And there are two hypothetical ways to do it. First, to treat people with dopamine itself, systemic. But dopamine is not permeable via the blood-brain barrier. And therefore, if you inject it systemically into the blood vessels, it cannot enter the brain because of the blood-brain barrier. And therefore, people use a precursor of dopamine, which is permeable via the blood-brain barrier, easily permeable. But you are quite right that, and this treatment is very efficient, but only for 3, maximum 5 years. And after that, L-Dopa becomes highly toxic. Well, and it's not really very clear what happens with this treatment, with this L-Dopa. But there is a lot of papers actually, just to understand how to improve this therapy, this treatment. But the most probable mechanism is the increase, the promotion of dopamine synthesis. And if there is no enough vesicles to capture this dopamine, it is accumulated in the cytoplasm, okay? And it becomes toxic. I repeat once again. If there is an increased synthesis of dopamine and this accumulation in the cytoplasm, in the cytosol of the neuron, and if there is not enough vesicles which are capable to inactivate this dopamine by its capture, it is accumulated in the cytoplasm and it becomes toxic.
So then excessive sport can cause Parkinson's.
Sorry?
Or not?
Yeah?
Excessive sport
Yeah.
the increase of this dopamine synthesis can cause or can be associated with the higher risk of Parkinson. Is it true or?
Yes, I told you that
Yeah, yeah.
at the beginning it is very useful for treatment, but after all, we're obliged, as the degeneration of dopaminergic neurons proceeds, we're obliged to increase L-DOPA
Okay.
dose. And at a certain dose, it becomes really toxic because of the reason that I explained already.
Thank you.
And how this toxic effect will demonstrated?
How this toxic effect
Yeah.
is demonstrated?
What, how patients feel under this toxic effect?
No, first it has been demonstrated in the animal models and in vitro models. Well, so and there are also, but usually we cannot really manipulate with people, with patients.
Okay, what how did those animals
How they
under
have they?
patient?
Well, so if we give dopamine, right, we have all the opportunities to study dopaminergic neurons after the dopa injection. We can actually we have a lot of facilities to study what is the concentration of L-DOPA in the cell body. What is the number of vesicles with vesicle membrane transport, etc. Well,
It's in vitro.
sorry.
It's in vitro.
Yeah, in vitro and also in animals.
Okay.
In animals. Those are the two kind of animal models. This is the general principle because we cannot step by step. We cannot study the pathogenesis of any disease in patients. We cannot treat them in the biological materials that is not easily available. That's always problematic, etc. But we can manipulate it anyway in animal models and also in cell models. But certainly we could imitate only certain processes, pathogenetic processes, not the pathogenesis as a whole. We don't know any model, any experimental model, which reproduces for 100% of any disease, could modulate only several processes. And in our laboratory, actually, we're trying to develop, to improve this L-DOPA therapy. But L-DOPA, after 3-5 years, it becomes toxic, and there is only a possibility to treat people. This is a surgical, functional surgery. This is an operation which is called high-frequency stimulation, like a pacemaker of the heart. Everybody knows, yes, about the pacemaker of the heart. Well, so the same pacemaker is introduced into the brain. Well, so the people, but I mentioned already, probably during last lecture, that there are dangerous side effects, particularly over the initial 2-3 weeks. There is high depression in patients because of this operation, and even suicides. Well, so that's not very easy as everywhere.
And how is toxic effect noticed in animals due to higher concentration of
Well, so in fact, what happens? The dopamine and the metabolic products, they become toxic for mitochondria.
Mm.
Well, so they destroy the function of mitochondria as a source of energy. And this is known in the literature as an oxidative stress. And the oxidative stress, this is the reason, the cause for the death of any cell, including neurons. Well, so finally, the effect of this toxic effect of dopamine is realized via the destroyed mitochondria and the loss of the source of energy, intracellular energy. Well, and I told you that peptide engineers are widely distributed all through the brain, all through the brain. And there are hundreds and thousands, I think.
Excuse me, didn't you say there were two reasons why dopamine should be separated?
Sorry?
Weren't there two reasons why dopamine should be separated? One, that it becomes toxic, and there wasn't a second reason.
That isn't exactly.
I just thought that in the beginning, when you started talking about dopaminergic neurons, you mentioned that there are two reasons
Yeah,
why it should be separated.
yeah.
And
Dopamine itself.
yes, yes.
Yeah.
One, that it's toxic when
Yeah.
it's not separated
Yeah.
and it doesn't all. And the second reason?
And the second reason, because dopamine, it can be released, it can be discharged from the neurons via exocytosis of these vesicles. Well, so after accumulation of dopamine in the vesicles, it becomes capable to be released via exocytosis of the vesicles.
Yes, thank you.
Good. Well, so I told you that there is peptidic neurons that distributed all through the brain, but this is not the case for monoaminergic neurons. And you can see that there is the only one source, the only one place of location of serotonin neurons in the so-called giraffe nucleus in the brainstem. Okay? But the axons of these neurons are distributed, are projected all through the whole brain, all through the brain. And therefore they provide serotonin, provides the control of the functioning of most brain regions. The noradrenergic or noradrenergic system is located also in the brainstem. And also its axons are distributed all through the brain. And they control almost all known brain functions. And this is also the case for dopaminergic neurons. But instead of, in contrast to serotoninergic and noradrenergic systems, there are two centers for dopaminergic neurons, for the accumulation of dopaminergic neurons. In the mesencephalon, in the substantia nigra, and these neurons control motor behavior. And these neurons are destroyed, are degraded in Parkinson's disease. And another place of accumulation of dopaminergic neurons, this is hypothalamus, and namely arcuate nucleus of the hypothalamus. And these neurons control the endocrine functions, neuroendocrine functions. Many neuroendocrine functions, but mostly reproduction. Because dopamine, which is synthesized in these neurons, is released into the hypophyseal portal circulation, and it inhibits the secretion of prolactin, hypophyseal prolactin, which is a hormone of reproduction. Yeah.
You said that in contrast with serotonin, noradrenergic system, dopaminergic system is not present in the midbrain, right?
No, I mean that in the serotoninergic and noradrenergic system, there are only one place for the accumulation of these neurons in the brain. This is brainstem, okay?
Yeah.
This is giraffe nucleus and this is locus coeruleus. And for dopaminergic neurons, there are two places in the brain for the accumulation of dopaminergic neurons. One of them, this is mesencephalon, and this is substantia nigra. It's not visible here, but in this region. This is mesencephalon and the substantia nigra.
And what about ventral tegmental area? Because I guess there are a lot of.
Ventral tegmental area. Well, so there are dopaminergic neurons, but in addition to dopaminergic neurons, there are many neurons which express only the first enzyme of dopamine synthesis. Well, so they are not dopaminergic in fact. Okay. Well, so and another center, this is arcuate nucleus of the hypothalamus. Well, Okay. Well, and look this here. That we know actually the dopaminergic neurons, which are located, as I mentioned, in the substantia nigra, in the mesencephalon, and also in the hypothalamus, and they contain a few of really functionally important molecules. I repeat just two enzymes for dopamine synthesis, dopamine membrane transporter, and vesicular membrane transporter type 2. It should be mentioned that the vesicular membrane transporter, this is also a characteristic of any other monoaminergic neurons, not only of dopaminergic neurons. This membrane transporter captured serotonergic neurons, and norepinephrine, and the dopamine as a precursor of norepinephrine is norepinephrinergic neurons. Well, so these molecules are specific, and this molecule is specific, but for all monoaminergic neurons. But in addition to these dopaminergic neurons, there is a population of non-dopaminergic neurons, but which partly express dopaminergic phenotype, which partly express the characteristic of dopaminergic phenotype. First, they synthesize, they have not both enzymes for dopamine synthesis, but only one of them. Either the first one, tyrosine hydroxylase, or another one. This is aromatic amino acid decarboxylase. These neurons, they have no dopamine membrane transporter. And these neurons, they have no vesicular membrane transporter. Well, so these neurons, they express only one of the enzymes of dopamine synthesis. Well, so before the end of 80s, people, they could detect dopaminergic neurons by using inside the neuron, by using immunocytochemistry for only one enzyme, tyrosine hydroxylase. But at the age of 80s, there was developed so-called double monolabeling technique. And at that time, we could detect not only one, but two enzymes at the same time in the same neuron. Understand? No? Well, so initially, because of the technique limitation, we could detect in the neuron only one enzyme, but not both of them. And therefore, people considered all the neurons which express tyrosine hydroxylase, they were considered as dopaminergic neurons. But at the end of 80s, the people, they developed so-called double labeling technique. And we could detect at the same time, in the same neurons, two enzymes, tyrosine hydroxylase and L-amino acid decarboxylase. And it happened that in some neurons, we could find only one enzyme, tyrosine hydroxylase, but not another one. And therefore, these neurons were called monoenzymatic neurons. You can see that was a pioneering work of which was made in Karolinska Institute in Sweden. Well, and that was very surprising. And the most advanced scientists from different countries, from Sweden, Netherlands, France, the United States, they combined their facilities just to understand what might be the functional significance of the neurons which contain only one enzyme of dopamine synthesis. Good morning, lady. well, and but they could not solve this issue. Huh? And they considered, just hypothetically, that there are only few neurons which express only one enzyme, and these neurons, they have no really important functional significance. And they have forgotten completely this issue. Well, it happens from time to time in science. What does it mean? No? Okay. Well, and at the beginning of 2000, we have returned in our laboratory, we have returned to this issue by using double immunolabeling of two enzymes within the same cell and with using confocal microscope by studying these neurons in the arcuate nucleus. And we found that in the arcuate nucleus, like in the striatum, there are three types of neurons. Green, which contain only tyrosine hydroxylase, red, which contain only aromatic amino acid decarboxylase, and yellow, which contain both enzymes. Okay? And therefore, we start from this point, we start to study the functional activity of these monoenzymatic neurons. And we have demonstrated that in addition to dopamine synthesis and dopaminergic neurons, by tyrosine hydroxylase and L-dopamine and decarboxylase, there is another type of dopamine synthesis by monoenzymatic neurons. Thus, in tyrosine hydroxylase containing neurons, L-dopamine is synthesized. And the L-dopamine is captured by the neighboring cells, neurons, which express only one decarboxylase for dopamine synthesis. In other words, dopamine is synthesized here in tandem by both neurons. And there was another question, what might be the functional significance of this so-called cooperative synthesis? Because certainly for many reasons, for energy supplying, etc., this synthesis is more efficient than this one. And we have demonstrated, first, that dopamine is synthesized by non-monoaminergic neurons in saturation, and then that cooperative synthesis of dopamine is a compensatory reaction on the failure of dopaminergic neurons. For instance, in neurodegenerative diseases. It means, well, sure, I mentioned last time that at the beginning, in the evolutionary or in ontogenesis, any cell can produce any physiologically active substance, serotonin, peptides, receptors for these substances, and the transporters. And after all, along the evolution of the organism, there is a depression, there is an inhibition of most synthesis. And the cells are specialized in certain synthesis. And therefore, for instance, peptide-agent neurons, they are specialized in synthesis of peptides. But under the local dopamine deficiency, these peptide-agent neurons, they begin to recollect that historically they can synthesize other substances. And they begin to synthesize, some of them, they begin to synthesize tyrosine hydroxylase and others decarboxylase. Well, so these monoenzymatic neurons, they are peptide-agent neurons. Well, so in addition to peptides, neuropeptides, they, under this deficiency of dopamine, they begin to synthesize, but they can synthesize only one enzyme. And therefore, dopamine can be synthesized only in tandem by two neurons. Yeah,
So you said that this is an adaptive mechanism in neurodegenerative diseases.
Yes, for instance.
And could it happen in healthy brain also?
In healthy.
Yeah.
Well, so this cooperative synthesis it can be realized in any situation which we need, the brain needs the increased functional activity, the increased amount of monoamines. And for instance, in acute nucleus, dopamine is synthesized in cooperation under the normal conditions. But under the degeneration of these neurons, this cooperative synthesis increases significantly. In the striatum, which is the target neurons for the target region for the regulation of motor behavior, in Parkinson's disease, in norm, there is no synthesis of dopamine. But in Parkinson's disease, under degeneration of dopaminergic neurons and the local deficiency of dopamine, there is many neurons, they begin to express individual enzymes of dopamine synthesis, and dopamine is synthesizing, but only in pathology, in Parkinson's disease. Well, and also we can see that L-dopa, which is synthesized in tyrosine hydroxylase neurons, now we know this L-dopa can play a role of an individual specific neurotransmitter. Well, very recently it has been demonstrated that the cells and the neurons in particular, they contain specific receptors for L-dopa. This means that L-dopa, which is produced here, it can play an independent role of a neurotransmitter, and it can be used for cooperative synthesis of dopamine. And L-dopa, it can promote the synthesis of dopamine and also of serotonin when it is captured into the serotonergic neurons. Well, so that was that finally the people from Karolinski Institute, they don't like me at all. Because they fail to understand what is the functional significance of these neurons, and we succeeded to do it. Well, so at the international meeting, during my reports, they're sitting like this. Well, and also, probably it is interesting for you that when I was younger, I was a PhD student. And the administration asked me to write an annotation of my contribution to science at this age. Yeah? I wrote 3-4 pages. Now I write 4-5 sentences, 5 lines. And this is one of these lines, because I think this is a really, let's say, great contribution, which makes it possible to understand, which discovered fundamental new principle of neurotransmitter production and action on the one hand. On the other hand, I consider, and it is actually accepted all over the world, that this is one of the most important compensatory mechanism or mechanism of neuroplasticity and the degeneration of the neurons. And it is most probably that it's not also only the case for dopaminergic neurons, but there are some reasons to think that this is also the case for serotonergic neurons. Well, so I think that discovery will be very, very intensely used, at least for recent 50, 100 years.
I have several questions. First one, considering L-dopa, so it does take part in this process with dopaminergic neurons, but probably I didn't get where it is synthesized itself, or I see L-tyrosine. So okay, how does it? I didn't get the process here. Isn't the same neuron of the not dopaminergic neurons? Not a type. I just okay. Let me put it this way. You've mentioned that the neuron is affected by several dozens of neurotransmitters,
Right.
and they at the same time, they are typicalized. I don't know. They
Classified.
classified by the dominant neurotransmitter or the speciality of it. Yes.
Yes, sure. It
Um,
depends on receptors.
Okay, then probably got it. I will check it later. And once again about this process of cooperative
Synthesis.
synthesis in healthy brain, it happens, but the where exactly and you and yes, you mentioned that it becomes more important during the neurodegenerative process. So, where in healthy brain this cooperative synthesis happens, or is it not clear, or
Right.
no?
Okay, I understand. Well, so in fact, you asked me what is the location of these neurons in the brain and what might be their functional significance in different regions of the brain. Well, so if first these people, Hochfeld, Thomas Hochfeld and Fuchs, they're really outstanding scientists in the Karolinska Institute. Well, when they say that most probably these are casuistic neurons, there are a few neurons which are not of important functional significance in the brain. Now we know that there are thousands of these neurons in the brain, many more than the number of dopaminergic neurons. Okay. First, then we know that they're distributed all through the brain, and not only the brain, but also they were detected very recently in the spinal cord. In the spinal cord. Well, so it means that the functional significance should be very sophisticated and very and of different kinds all through the brain. It's not started yet. Therefore, I said that this is an issue for the study for the next 50 or even 100 years. But what is also important in this connection, that on the one hand, this dopamine, which is synthesized, and N-dopamine, which is synthesized in so-called monoenzymatic Th neurons, and dopamine, which is synthesized in cooperation. They can play a role of a neurotransmitter, okay, providing their action on the neighboring or the next neurons, target neurons. But there is another very important issue, that these neurons project their actions. They're located very often around the ventricular system of the brain. Well, so the brain, it has its own system for circulation of the body fluid, which is called the cerebrospinal fluid, inside the brain. Well, so the brain, I will tell you a little bit later, that the brain, it has its own endocrine system. In the body, we know an endocrine system consisting of endocrine glands, which are secreted into the general circulation, and these substances are distributed all through the body. But the brain, it has its own endocrine system, and the brain is protruded by so-called cerebral ventricles with a circulation of so-called cerebrospinal fluid. And these neurons are very often allocated around these ventricles, and they project their actions into the ventricles, into the cerebrospinal fluid, secreting their products into the cerebrospinal fluid. Well, so it means, well, and after all, these substances are distributed all through the brain, and they are released into the other brain regions, and they provide not only neurotransmitter but also neurohormonal effect on the target brain neurons. not only in the brain, but I mentioned also in the spinal cord. Well, so even 10 years ago, even 5 years ago, we couldn't imagine that there are the same neurons even in the spinal cord. It has been discovered by, as far as I know, by Chinese people. But still, I published 2 review papers on this issue, and I told people they cited only individual papers which are devoted to this one enzymatic neuron. They cite at least 5-10 papers which are delivered from our laboratory. Well, so that is, I told you again, that this is one of the lines in my contribution to science, a real contribution. Well, okay, where's no one? Good. And last time somebody asked me a question, whether we could do a section, whether we could study the interneuronal relations of between different neurons, monoaminergic, peptidagic, and others. Huh? And I just give you an example of such a study. Well, so the final physiological effect of any neurohormone, of any neurotransmitter, this is a resolution, a resultant of the action of the whole neurotransmitter neuronal network. Okay. For instance, this is a network of the neurons for the regulation of peptidagic neurons in the hypothalamus. Look, please, these neurons are under the control by the neurons of the brainstem, usually serotonergic neurons, roughly those, okay? There is under the norepinephrinagic neurons, under the other peptidagic neurons. Well, so the final resulting action of these neurons is a result of the action of a number of neurotransmitters. And nowadays, we have special techniques to differentiate, to identify, to visualize these neurons. Look, please, these are neurons, red, yellow, green, and these are neurons which contain different neurotransmitters and different specific enzymes. Well, some of them are monoaminergic neurons, some of them peptidagic neurons, and we can see, and we could study the interneuronal relations between these neurons. When using electron microscopy, we could study even synaptic contacts between these neurons. But this is at the light microscopic level. Yeah?
If the neurons of different types, for example, serotonin and dopaminergic neurons connected to each other, does it mean that the dendrites of a particular type of neurons can receive the neurotransmitters of the other type?
Certainly. Well, so, and I will tell you, when I will tell you about neuroplasticity, I will tell you that these neuronal networks, they're not stable at all. Huh? They, in accordance to the changing in the functional state of the brain and the whole body, these networks are reorganized and very briefly, in a short time. For instance, I told you that synaptic contacts, they can be created and can disappear within the minutes or even seconds probably. This is a very, even 20, 30 years ago, we have considered that these interneuronal relations are very conservative and they are very stable, but that's not at all.
Question. This in the upper right corner picture, it was by fluorescent.
Yes.
Okay.
That was stained. These neurons are stained, normally stained with antibodies to specific antigens for proteins or enzymes for monoamine synthesis, or even for monoamines. And these antibodies are labeled with fluorescent markers or fluorescent stains. Okay? I will tell you a little bit about immunological methods a little bit later. Is it over? Okay, good. Good. Don't worry, I will give you all the materials until the end of our course. Okay, you're free. Very good. It's a long break.